According to the Global Cancer Statistic 2018 by the World Health Organization, in Malaysia, there were 20,619 new cancer cases for male and 23,218 new cancer cases for female. The number of cancer-related deaths for male were 13,937 cases, while for female, there were 12,458 cases. This made up to 26,395 cancer mortality cases. According to the report, the top 3 most frequent cancer in Malaysian males were lung cancer, colorectal cancer, and prostate cancer, while the top 3 most frequent cancer in Malaysian females were breast cancer, colorectal cancer, and cervix uteri cancer. In 2018, there were 7,593 Malaysians who were diagnosed with breast cancer, and 6,137 Malaysians who were diagnosed with colorectal cancer. Cancer starts from carcinogenesis process, whereby normal cells are transformed into cancer cells. This transformation involves mutations at cellular, genetic, and epigenetic levels. There are 4 stages of carcinogenesis. First, tumor initiation, where a chemical carcinogen causes a genetic damage by modifying the molecular structure of DNA, which leads to a mutation. Second, tumor promotion, where the initiated tumor cell goes through selective clonal expansion, producing a larger population of tumor cells. Tumor promoter agents, such as croton oil and benzoapyrene chemical, contribute to this process by reducing the latency period of tumor promotion. Third, malignant conversion, where the tumor cells transform into malignant cells. Fourth, tumor progression, where malignant cells acquire more aggressive characteristics over time, such as tendency of genomic instability and uncontrolled growth. When DNA damage occurs, it will trigger the activation of tumor suppressor genes. These genes arrest the progression of cell cycle, conduct DNA repair, and prevent mutations to be passed to daughter cells. Ultimately, it is the accumulation of DNA damage without repair that leads to cancer. One of the most studied tumor suppressor gene is protein 53, p53. This protein carries important anti-cancer functions in our body, including apoptosis, programmed cell death, genomic stability, and inhibition of angiogenesis, tumor blood vessel growth. Cancer is characterized by deficiency of apoptosis, leading to malignant cells that don't die. Therefore, medicines capable of restoring apoptosis holds a key into the future of a safe yet effective cancer treatment. Txl phytoceutical, which compose of 14 herbal ingredients, has been shown to induce apoptosis in a wide variety of human cancer cells. The research published in the Alternative and Complementary Medicine in 2005 by Andy Sun, PhDs, and others examines the apoptotic effects of Txl on 15 human cancer cell lines and 2 normal human cells. The study used tunal assay to investigate this apoptotic effect. The result demonstrates that cancer cells originated from 15 human organs all underwent apoptosis, tunal positive, while the normal HUVEC and PBMC normal cells did not. These data confirm that Txl has a therapeutic effect on a broad spectrum of cancers, including human cervical carcinoma cells, C33A, lung carcinoma cells, H1299, and hepatocellular carcinoma cells, HepG2. Txl phytoceutical also inhibit angiogenesis and metastasis of cancer cells. Jean Sanchia and others investigated the effects of Txl on neovascularization of HMEC1 cells and HUVEC cells in the presence of 10% FBS angiogenic stimulator. The result shows that Txl significantly inhibited the tubular formation of HMEC1 and HUVEC in a dose-dependent manner. To test the effects of Txl on cancer cells' motility, Chia experimented the movement of 4 highly invasive cancer cell lines, including PC3, human prostate cancer cells, MDA-MB231, breast cancer cells, H1299, lung cancer cells, and CT26, colon cancer cells. The cancer cells were allowed to migrate into the gap created by scraping, either in the absence or presence of Txl. After 20 hours of incubation, the gap of cancer cells migration in the Txl group was wider than the non-Txl group, indicating that Txl inhibited the motility of these 4 invasive cancer cells. Together, this study concluded that Txl can inhibit cancer metastasis and angiogenesis by targeting multiple biological and pathological processes in cancer cells. In conclusion, cancer is characterized by insufficient apoptosis. Tumor expansion occurs as tumors induce its own blood vessel growth, angiogenesis. In the overall stage grouping, metastasis places a cancer in stage 4. Txl is able to induce apoptosis in a wide variety of cancer cells, inhibit metastasis and angiogenesis, suggesting that Txl has obvious therapeutic effects on a wide variety of human cancers in a dose-dependent manner.